Welcome to the 54th InterScience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC, and this is ASM Live. I'm Michael Schmidt, and together with the help of the program committee, we've selected some of the papers that are being presented here this week for a discussion. And joining me today are Kieren Marr from the Johns Hopkins University and Andrew Ullmann from the Julius Maximilian University. And today we're going to have a discussion on new antifungal options for cancer patients. They're principally going to discuss a newly developed antifungal, isavuconazole, which was found to be effective as an existing drug, voriconazole, against invasive mold disease in cancer patients with less adverse effects. We all recognize that any antimicrobial has side effects, and this one had fewer than the current drug out in the marketplace. The researchers are going to discuss their exciting data that are going to be presented tomorrow, so you still have an opportunity to go and chat with the investigators at their posters or talks. And I encourage you, the audience, and those of you following us on the chat line, to come with questions, and we're just going to have a discussion about this new and exciting field of developing antifungals for the use in the clinic. So welcome, and thanks for agreeing to do this on a Sunday morning, and we're delighted to have you. So Dr. Marr, why don't you start off and tell us a little bit about the background and the study that you're going to present tomorrow.
Sure. First, thanks for inviting us.
You're welcome.
Exciting to be here, even on a Sunday morning. The studies that we are presenting tomorrow will describe the results of a randomized trial that was done to analyze the efficacy and the safety of treating invasive mold infections, which are usually infections that occur in the lungs in people who are severely immunosuppressed. And the study was done worldwide to compare a new drug called isavuconazole, which is in the triazole type of antifungals, compare that to our standard drug that is currently first line for invasive mold infections called voriconazole.
So was this a placebo-based trial, or was this an inferiority type of trial?
So it's not a placebo-based trial. These patients need therapy.
So ethically it's not responsible to do a placebo-based trial?
No. The outcomes would be devastating. That's not the thing to do. So this is a randomized study to compare non-inferiority with one drug compared to the other. And I should add that tomorrow we're presenting the results of the subset of patients, the majority subset of patients that had hematologic malignancies as their risk for their mold infections.
So these people who have these hematologic risks are on significant immune suppression as a consequence of the chemotherapy that they're enduring, making them very susceptible to fungal infections, especially the ones that become invasive.
Exactly. It's a rather common complication of receiving chemotherapy or even a bone marrow transplant for underlying leukemia or other kinds of hematologic malignancies or blood cancers. So we have worked for now a couple decades in trying to improve our therapies as well as our diagnosis, and this is the newest drug that is available.
Dr. Ullmann, what about you? What are you going to teach us tomorrow?
Well, basically it goes to the same direction. We're looking at different subsets, and the subset I'm presenting tomorrow is uncontrolled cancer. We do know that patients that have a new diagnosis of cancer, this would also include leukemia patients, or have a relapse and go through chemotherapy, they're severely immune compromised, and again they are as well endangered for fungal diseases, and if they develop a fungal disease, especially filamentous fungi, they have a likelihood of death, and that's why we really need these new drugs out there that have to be evaluated for.
So let's talk a little bit about new drug development for fungi. What does this particular drug target?
So this, as an azole compound, like the other ones, targets the production of ergosterol in the cell membrane, which is a critical sterol, like in our cell membranes cholesterol is. And so you were right in describing fungi very similar to us, but there are some differences, and one is the plasma membrane and the sterol components.
And so I cheated and looked ahead to see what you were going to present. So one of the interesting things is in your abstract, you specifically talk about fewer side effects. So what sorts of side effects do these classes of drugs, the azoles, specifically deliver to the patient? And obviously I gave away the punchline. This new drug actually had fewer side effects. So why don't you share with us that, so individuals watching us or those in the audience that may not know can understand why this is a take-home event.
Okay. Well, so all of the drugs, essentially all of the drugs in this class have a primary side effect of having liver toxicities, and they're largely metabolized by the liver. Years ago, when voriconazole was approved, it was approved by comparison studies to amphotericin B, and that was an even more toxic drug with kidney toxicities. And that was really a major advance for treating invasive mold infections. That comparator drug, though, since we've been using it for now quite some time, we've learned that this drug as well has some very unique side effects. Not only the liver toxicities, but a peculiar photosensitivity rash, and a rash that can actually heighten risks for skin cancers if someone's on the drug for a long enough time. The other toxicities include visual changes and some hallucination type of side effects. And so there's been some controversies as to how important that is. It's certainly important if you're the patient that's experiencing these things. And so the results of this study really showed that there were relatively fewer of those kinds of side effects, ones involving the liver, others involving the eyes, and neurologic hallucination types of side effects, as well as the ones that we can see pretty frequently with regards to skin side effects and rash.
So the rashes and the photosensitivity to being out in the sun and things along those lines were also reduced.
Exactly.
So Dr. Ullmann, what about you? What's your magic take-home element?
Well, maybe I might add something what Kieren just said. When you look at our posters, you see something, and I'm crunching the numbers now a little bit. I think it's also important to realize those numbers. If you look at the treatment emergent adverse events in both groups, it is significantly high. It's high in both groups. It's just all the adverse events that are being just reported during a study. And this study is a double-blind study. And the investigator reports all adverse events, and that's, of course, good clinical practice that's necessary. But they also have to evaluate these adverse events. And if those adverse events are evaluated by the investigator saying, okay, this is treatment related to the drug, the study drug. Then
or as opposed to their condition.
Exactly, because the adverse events that we are registering, it's around the 98% margin, so it's very high. It's somehow confusing that we're saying, oh, there are less adverse events. But if you look at the design of the study, which is double-blinded, meaning the investigator and the patient do not know what kind of drug they're receiving, basically the evaluation of the adverse event is unbiased, because after the study is finished, it's going to be unblinded and evaluated. And that's the point where we really saw the significant decrease of adverse events that we usually see with liver toxicity, visual or hallucinations, skin toxicities that were significantly lower with isavuconazole in comparison to the comparator drug.
And that's a fine example of the limitations or the positive aspects, depending on if the glass is half full or half empty approach to looking at inferiority trials. First and foremost, you can do the double blind because you have the ability to give an ethical treatment to a patient, And you then will actually be able to tease out from the data whether or not the drug is indeed better.
Right. And that's what we basically did. We worked together on the data review committee, and we actually evaluated all those patients receiving both study drugs, seeing what the outcome was, and of course the primary endpoint of the study was all-cause mortality. And that's a very hard endpoint that cannot be debated, dead or alive, at the end of the story. And basically the rates of all-cause mortality was equal in both groups, but again, at least in my group with the uncontrolled cancer, it was overall higher than those with not uncontrolled cancer. So that really gives us the picture and confirms the picture that those patients with cancer or leukemia or allogeneic transplantation or so whatever, that they are still at high risk of death despite adequate treatment. So that's why we really need more to help our patients. More
drugs or more studies?
All of the above. All of the above, of course.
You know, it's a funny way of looking at it, but it really drives home the need for new antifungals because you know, the targets are much more limited in the eukaryotic side of the coin as opposed to the prokaryotic side because we don't have peptidoglycan, the wonder, the true gift that the prokaryotes gave mankind is, I think, always been peptidoglycan, given the number of beta-lactamase antibiotics that are out there. So, where to next? So you've begun to evaluate this drug. You found a good indication. So there's always a what's next. Where do you see this actively being moved into the clinic based on the results from your two studies? Is the company that's manufacturing the drug going to seek FDA licensing or the European equivalent? So I guess that the selfish perspective for the patients are, when will I be able to have a physician prescribe this drug so I get fewer side effects and get better faster?
Well, obviously we're not the sponsor of this study.
That's right.
So, but I would like always to refer to the sponsor, but there was a press release, I think a day or two ago, from the sponsor actually indicating that the drug was now submitted to FDA for the, I think it's called Padova. So it will be now evaluated by the FDA within the next 6 months. I think that's the procedure here in the US. So that by the first quarter of next year, there'll be a result coming back from the FDA. And the equivalent with the EMA in Europe, there was a press release by the sponsor as well, which is now submitted for the MAA as it's called. And there should be some results back by the end of next year that actually also shows how slow things have to develop to get this drug into the market from the beginning of the study until getting it market ready takes a while. And that's always a challenge for our patients, of course.
Yeah, you know, I think the press release described that the FDA accepted the new drug application and its evaluation for a potential expedited review under an orphan drug for aspergillosis or mold infection. So I think that that will probably be evaluated soon. And I think based on this large study, there's a robust amount of information.
There's enough patient data in the mix for the FDA to make rational determination and the equivalent regulatory body in the European community as well. Right.
Well, yeah, I can't speak to the review process of the FDA, but I think that it's a rather robust data set with over 500 patients exposed in this particular study.
We have a question from the audience, it looks like.
Yeah, good morning. It's Michael Smith, MedPage Today. A couple of quick ones, if I may. The AE profile, you said there are fewer. What about, so that's numeric, maybe you can throw some numbers at me. That would be nice. Because you have two separate, slightly separate subpopulations here. But, so I don't know how you can maybe tease them out in some way, but I'd like to know sort of what the numbers are like. And then the other question is, assuming that a patient, regardless of drug, gets one of these side effects, are they milder in the new drug arm, or are they the same type, just there aren't as many of them? That's the first one.
Can you start off with the uncontrolled cancer patient population? And I would also like to advise to look at the poster. There are handouts there that you get all the numbers in there. But the percentage of those patients with isavuconazole drug-related adverse events was 39% versus to the comparator was 61%. And that difference came up with a p-value less than 0.05, which is considered significant. That's at least to the part of my trial. I don't know if you...
So, and I think it's important to be clear as to what numbers we're referring to here. And just to go back, the results of the entire study was presented last May, I believe, or June, at ECCMID by Johan Martens. And so that was the large data set that was really the large safety set that was from the ITT analysis. And, you know, almost, I think, 100% of patients on this study had an adverse event that was reported, and that's characteristic of the sickness and their underlying conditions. But the real difference came in the reports of treatment-related adverse events, or those events that were thought to be related to the drug. And I think that in one way it was because of the double-blinded nature, of course, blinded as to who was getting what drug. I would, however, I pause when I look at these results because you know, they are informed by what we know about the drug-related toxicities of voriconazole. And it's been almost a decade of use of that drug that has enabled us to understand the skin rash and so forth. And so you can almost anticipate an increase in reporting of those effects that we anticipate to be related to voriconazole. We don't know if there are peculiar drug-related toxicities to isavuconazole because we've not used it as much as voriconazole. And so I think that, you know, that those are kind of pieces of information that come when we have the drugs available after phase 4 and more widespread use. But there was significant differences. Of course, the study's powered to show differences in overall, or the similarity, non-inferiority in overall survival. Mm-hmm. But when you compare the two groups, there were clearly numeric differences and substantial numbers on the order of 20% plus with regards to the rashing, the visual changes and so forth that you would expect with voriconazole.
So the differences for the individual ones are roughly 20% between the arms?
Please come to the poster tomorrow. If you look at the overall study... No, I'm writing this today. Okay. That's my problem. That's why I'm asking for data rather than sort of broader statements. I don't have the numbers in my head right now, but I could come up with them by the end of today. All right. What about the mildness? If you get a rash with the new drug, is it the same as the rash with the old drug, or is it in fact milder? Well, that we don't know. The severity, so when we say treatment-related adverse events, there were more severe, treatment-related severe adverse events. And so it's on the typical grading scales, one could presume that these were severe events if they were high-grade, severe adverse events. But we don't have a quantity of rash on one versus rash on another. We didn't grade those.
That's probably a different study. Okay. Another question from the audience.
I'm Jack Bennett from the National Institutes of Health in Bethesda, Maryland, and I have a question about the blood levels of active drug with isavuconazole. And the reason I asked this was the older drug, voriconazole, two patients getting the same dose could have a 4 or 5-fold different amount of bioactive drug in their blood. It'd be nice if isavuconazole was more predictable, like the even older drug, fluconazole, was so predictable. So what about the variability of blood levels with the same dose as isavuconazole?
Dr. Bennett, thank you for that question. That's outstanding. To our guest here,
I'm trying to crunch the numbers again, what I have up in my head. And if I recall it correctly, because there's still the analysis still ongoing, especially for the ECMIT presentation from last year, that was pretty good levels in regards to the exposure of the patients. I know that. But your question was more precise. Was there inter-individual variation? Did we see anything to that? I have to actually look it up. It would be not very professional if I give you my guess right now. Sorry. I don't know if you know anything better.
No, I haven't seen those data come out of this particular study right now. And there's actually 18 posters here at this meeting on the drug and various different analyses that come largely out of this trial as well as other sub-studies. So one would anticipate, and this is a very, very important issue, because the drug levels of voriconazole, as many know, are so unpredictable probably because of metabolism and variation in metabolism, that we are largely encouraging people to use therapeutic monitoring of levels, especially to decrease the amounts of toxicities that one might see. And so that was not incorporated into the trial routinely, and that is potentially a significant variation from how we practice currently. Of course, at that point when the trial was started, that wasn't done widespread either, and so this is kind of an indication of how we've changed over the last 5 years as well. Isavuconazole, my understanding is that it's actually administered as a prodrug that spontaneously breaks down in the serum to isavuconazole, and there are some differences in metabolism, I believe, with regards to, for instance, 2C19, which is the problematic liver enzyme for voriconazole, and having some variation in drug levels. But that's a very important point.
Thanks, that's helpful.
Thank you.
It will also change the way patients are cared for if the blood levels vary from patient to patient based on their genetics or their activity of their liver, and this will probably become especially important in transplant patients who are receiving new livers, and you have no way of forecasting what the new liver or how the new liver is going to behave in the new patient, in the new recipient. And you know, it's that always double-edged sword of when you implant an organ before it's fully tuned up to its wild type, if you will, levels, and then the whole conundrum of humans not being mice, and we're a very outbred group of individuals, and consequently we don't have all the same liver markers, and it makes for a very challenging trial design.
Also, it's not only the genetics or a new liver, it's also the interaction. If you look at a transplant recipients, they receive tons of other kinds of medications, and usually it's also the same target in the liver where it's being metabolized, and there's some substrates or inhibitors of certain isoenzymes, which makes monitoring of a new drug that has a potential of toxicity very important. And we do have a lot of experience with the old azoles, but limited, of course, with isavuconazole. But to that regard, there are a couple of posters, and Kieren alluded to this already, looking at interactions with isavuconazole with other drugs and what the effect is on the metabolism and the exposure in patients with those other drugs. So I could encourage everybody to look at the posters. There are plenty of isavuconazole posters at ICAAC to review those pharmacokinetic data.
Or either that, or I need to get a bigger couch.
That's the other alternative, of course.
Is get a bigger couch to bring all the other authors on to have this discussion. We have another question from the audience. Thank you.
Hi, John Otromke, the Pharmaceutical Journal. Just wondering, does this new drug bring any new side effects or any kind of new adverse effects that were not seen in prior treatments? New side effects.
That's
a
great question. Great question, and we don't know. None that I know of would be probably the most accurate answer.
At least the numbers didn't show up in comparison to the other drug. That's for sure.
You've been...
And maybe let me just add one thing to it. We're talking about adverse events. There are two kinds of adverse events that we have to evaluate. These are treatment emergent, meaning all those adverse events are reported, independently if it's drug-related or not. And when you look at that large list of adverse events that are registered there was no numbers that were different except for those numbers that we were aware about that had typical, what you said very nicely. We know the other drug very well, and we know where to look at, and the numbers were different.
Yeah, I think that's one of the challenges associated with these inferiority trials is, you know, you're giving a drug, and you know the caregivers know the side effects associated with the drug that you're comparing it to. And given that it's double-blind, you almost have a bias of looking for those side effects when you almost need a double-blind test in order, you know, here's an antifungal that you're giving, but that's not ethically possible as well.
Yeah, I think that this is an important point. I think that the other important point is that these patients, by nature of who they are in the hospital with cancer, they're very complex. And so when we talk about the percentage of people who have adverse events reported, I mean, the adverse events are also related to their underlying disease and are usually related to the underlying disease. So it takes quite a signal to really appreciate a drug-related adverse event over the top of their adverse experiences from their usual therapy.
Which is, again, one of the challenges associated with developing, in this particular case, a new antifungal. Again, considering that this is not going to be administered to a general medical population, it's really being administered to a very special class. Fortunately, for the most part, humans don't get fungal disease, systemic invasive fungal diseases, and so it makes that management so much more challenging, and the development of those drugs so much more challenging for pharma that's looking at, you know, all the nuts and bolts associated with, do we develop a drug? So I'd like to thank both of you for our discussion here this morning. You've been listening to ASM Live at ICAAC, and I'd like to thank the audience for their participation here this morning. Please join us again at noon today. I'm Michael Schmidt from This Week in Microbiology, and please listen to me and my colleagues, Mozelio Schechter from Small Things Considered, Michelle Swanson from the University of Michigan, and our host of This Week in Microbiology, Vince Racaniello, as we conduct another edition of TWIM, the podcast that discusses the unseen life on our planet, Earth. Thank you, everyone, for your participation, and come back and join us again at noon. Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.